Okay. Well, good morning, everyone. We'll give a few more minutes for those who are hopefully going to join us today. Okay. All right. We'll give a few more minutes, perhaps. Andrew, maybe a couple of minutes or so, and then we'll get going. Um, okay. Well, we're going to get started. So Andrew can get started. Good morning, everyone, or good evening, good afternoon, wherever you're joining us today. Um, Permission brain tradition. If you can just type in the city, the state, and the country where you're coming from in the chat, so we know where you're all joining us today, that would be wonderful. And you can go ahead and do that now. If anyone there, there you go. New York, Nepal. All right. Well, while we're having you, Jacksonville, Florida, Cape Town, South Africa. Fantastic. Okay, while some of you are getting the hang of that, I'm going to get us started here. We are very happy to have Dr. Andrew Hale join us this morning and be the first in our lecture series for Mission Brain. Just to give you an introduction, Dr. Hale is also one of our Lisa Hannigan scholarship awardees. He's got a very long and illustrious career so far. Dr. Hale is a neurosurgery resident at the University of Alabama at Birmingham. He earned both his bachelor's and master's degree in biochemistry from Case Western Reserve University in Cleveland, Ohio, where he studied the role of transcription factor KLF4 in tumor angiogenesis and Notch signaling. He then matriculated into the Vanderbilt University School of Medicine Medical Scientist Training Program under a full tuition scholarship to earn both his MD and PhD degrees. Hale earned his PhD under the tutelage of John D. York, PhD, a Howard Hughes investigator, where he applied biochemical, molecular, mouse model, and genetic approaches to study the roles of lithium-sensitive phosphatases in cell signaling and disease. He was supported by an NIH F30 during this time. He then pursued a brief postdoctoral fellowship in human genetics and functional genomics at Vanderbilt, where he became interested in elucidating the genetic basis of neurosurgical conditions. He then matched into neurosurgery residency at the University of Alabama at Birmingham, where he is currently a PGY 5. Dr. Hale is currently completing a postdoctoral fellowship at the University of Cape Town Neuroscience Institute, led by Graham Fagan, where he is using brain organoids to understand the mechanistic basis of both genetic and acquired forms of hydrocephalus. He enjoys close collaborations and mentorship by Dr. Chris Cal of Massachusetts General Hospital, Harvard Medical School on the genetic and mechanistic basis of neurosurgical conditions. Dr. Hale's clinical interests include pediatrics, cerebrovascular, epilepsy, and global neurosurgery. His overall goal is to expand the use of genetic and molecular information in the care of pediatric neurosurgical patients. Beyond work, he enjoys traveling, wildlife conservation, fly fishing, and spending time with his wife, Haley. We welcome all of you for Dr. Hale. Thank you for joining us and for giving us the generosity of your time and expertise this morning.
Thank you. I really appreciate the invitation. Let me see if I can get my screen full. Okay, can everyone see my screen?
Yep.
Okay, great. So, um, so thank you so much for the Mission Brain Foundation for the opportunity to give this lecture. I've been very fortunate to receive the scholarship from you guys, and it's been absolutely critical to supporting my time here in Cape Town. So, I'm really interested in understanding kind of the molecular and genetic basis of disease, and I think that that really ties in nicely with hydrocephalus, and I'll kind of share a little bit as to how I got into this. So, my real passion is, really being at the intersection of basic science, genetics, and neurosurgery. Through a combination of this interest, I sort of stumbled into global neurosurgery, really just out of my kind of innate curiosity to understand disease mechanisms. And so I'll kind of talk to you about that a little bit and a step the stage. So, if you put in Chat GPT, what is global neurosurgery, you get this list of terms. And so all of these are incredibly important concepts, playing a role in, clinical care, delivery, and implementation globally, your health equity, surgical systems, education, sustainability, infrastructure, etc. You know, in addition to navigating challenging political landscape and country-specific problems or in improving kind of boots on the ground clinical neurosurgical care. But how I was thinking, how can I fit into this and where do I come in? So, my strengths and my passion is really in basic science and at the lab. And so when you think about global neurosurgery, you see a very different pattern of disease in many cases in different parts of the world that we don't see in the US. And moreover, our international colleagues, I think, oftentimes have really unique perspective in seeing some disease kind of at each end of the spectrum or each end of the bell curve. And so what I noticed as well in the schematic was that kind of understanding basic disease mechanisms is lacking. Me as an individual provider can only take care of, X number of patients, but if you can implement a new type of treatment or therapy that can then be propagated, across the world, I think that means you can kind of expand your impact. And so, I think many people kind of have this picture in their mind when they think of global neurosurgery. And, that was certainly my impression for a long time as well, especially being in hydrocephalus and pediatric neurosurgery. You can't go very long in the field without hearing about the work that Ben Worf has done in Uganda. And what's nice about this picture is that this is, kind of serendipitous. This is, Rob Naftal, as you can see here, who's a faculty member at Vanderbilt, one of my first neurosurgical mentors, and one of my current residency mentors, Jim Johnston, along with John Magumba, who has close ties here in Cape Town to Grand Vegan. And so I just wanted to highlight that here, but also kind of to really bring home the point and emphasize that global neurosurgery, I think, is much more than that. So how did I kind of get on this path? So, I think that there's kind of a serendipitous kind of explanation to this. So my mentor here, one of them, Dr. Jeff Blount, invited me out to dinner right before the beginning of the AANS in 2022. And shown here is Graham Fegan, who's the director of the Neuroscience Institute at University of Cape Town. And here's one of his clinical partners, Yico Enslan. And then back there is Rick Boop, who many of you will know. And he took me out to dinner, and we got to chatting and kind of talked about, University of Cape Town and what's going on there, and really emphasized the new development of the Neuroscience Institute, which I'll talk about. And then, Graham actually ended up inviting me to be, a visiting scholar or visiting professor to give a talk in Cape Town in early 2023. And this really kind of set the stage for what kind of my postdoctoral fellowship was going to be about. And so, Cape Town and University of Cape Town was a remarkable place. So it's home to here, which is the Red Cross War Memorial Children's Hospital, one of Africa's leading pediatric hospitals. That trains a significant proportion of the country's pediatric neurosurgeons. A lot of them can trace their lineage back to Red Cross, as well as Gutsy Hospital, which is where Kristen Barnard was performed the first human heart transplant. And so at the historic intersection of these amazing clinical institutes is the University of Cape Town Neurosciences, which is a multidisciplinary and integrative scientific and clinical center that is really focused on fostering an ecosystem of collaboration, innovation, and and here's a picture of Dr. Pagati operating here at Red Cross. And then here's a really nice picture of the upper campus that UC Food. And so many, worldwide kind of lists and rankings actually place University of Cape Town as well the top ranking university in the entire Southern Hemisphere. And so a little bit more about the Department of Neurosurgery here. So this is Graham Fegan. He, one of my mentors here, and he has, long been a huge advocate and voice in global neurosurgery. And he's the first director of the NI. And then here is Tony Sagagi, who's now the chief of pediatric neurosurgery at Red Cross, and also the scientific director of the NI. And so Tony's very unique in that he is an incredible clinical neurosurgeon, but also has a extremely active research group, which he's probably the world's leader in tuberculosis meningitis, as well as traumatic brain injuries. And he's been the recent recipient of a Wellcome Trust career award, which is on par, if not more prestigious than something like an NIH Director's Award. So I'm very fortunate to be, mentored by these two individuals who really kind of are at the forefront of science and neurosurgery across the world. And so I think that global neurosurgery is more than, what people think of kind of delivering care to impoverished regions of the world where, you're really just trying to make, an impact by, patient by patient. I think by studying some of the mechanisms and basis for disease that predominantly affect patients in low and middle income countries that we can treat with neurosurgery, I think is absolutely critical. And so a little bit about kind of the disease that I'm interested in. So these are scans of children with hydrocephalus. And this is a complex, enigmatic, and multifaceted problem. So hydrocephalus is a failure of brain homeostasis. It's a brain cerebrospinal fluid interface, which often leads to cerebral accumulation of ventriculomegaly and can cause elevated intracranial pressure. There's a number of different causes for hydrocephalus, ranging from genetic causes to infection to intracranial hemorrhage or congenital neurosurgical. And without treatment, hydrocephalus can lead to severe functional impairment or even death. And treatments are, limited to suboptimal surgical approaches where stunts often fail. 50% of stunts will fail within the first 2 years of their placement. And so also while lack of access to neurophysical care may be fatal, many patients with hydrocephalus also require multiple surgeries. And this will place a significant strain in the child's development. But importantly, no, uh, no pharmacological solutions have been proposed or utilized in Hydrocephalus.
Hey, Andrew.
So, yeah,
Sorry to interrupt you for a quick second. Your audio seems to be a little sounding like a little bit like you're in a bubble or something, and when you move, it makes a difference. So before you keep
okay,
going, because I know that a lot of us really want to listen in, I want to
okay,
bring that to your attention.
okay. Thanks for letting me know. Is this a little better?
Yeah. Try that again.
Okay, is this a little better now?
Yeah. Okay.
Okay. So, yeah, no problem. Okay. So here are scans of multiple patients with hydrocephalus. And so what you can see is there's highly variable neuroanatomy and highly and likely, or there are kind of all of these patients underwent highly variable clinical courses. And so obviously we're dealing with different phenotypes with very different etiologies of hydrocephalus, the age of diagnosis, neuroimaging features, and intertwined with normal brain development. And so, you know, there's a tendency, I think, to think that this is one disorder that is treated with a single surgical solution. But as we start to understand more about kind of the mechanistic basis of the disease, we can really see that's not the case. So, what is really the scale of this problem? So, South Africa, specifically, representative kind of sub-Saharan Africa more broadly, disproportionately impacted by hydrocephalus, which accounts for, this sub-Saharan Africa accounts for approximately 50% of cases globally. And this was largely due to neonatal and early childhood infection. And so what you can tell is that Europe and most of the West have the lowest incidence of hydrocephalus, yet the majority of patients are impacted in the southern hemisphere, either in Africa or South America. And in Africa in particular, there's only 1,900 neurosurgeons serving a population of 1.2 billion people. And so while expanding, the global neurosurgical clinical workforce is essential, I think that understanding the mechanisms underlying the disease, which could potentially lead to a new pharmacotherapy, would be potentially more impactful. But elucidation of drug targets, anchored in human biology and representative of the people that they would ultimately be treating is quite a daunting challenge. And so here is a map published recently last year in Nature Communications. And what this map shows is this ethno-linguistic diversity in Africa. And so Africa harbors the highest diversity, genetic diversity of anywhere in the world, with over 2,200 unique genetic diaspora. And diaspora essentially just means genetic founder pocket. But Africa's genetics are extremely underrepresented in global databases and drug discovery pipelines. And in fact, one of every 12 admissions in South Africa is directly due to an adverse drug reaction because many of the drugs that have been implemented for a variety of conditions, antibiotics, antihypertensives, you name it, are being developed and tested in populations that are in the global north. And so if we're to try to understand and tackle a disease like hydrocephalus that disproportionately impacts many African populations, we need to understand the disease in the context. And so, specifically, even in South Africa is the rainbow nation. It has over 13 official languages, and it also has one of the highest rates of tuberculosis in the world. So when you start to think about kind of the scope of the problem, it becomes quite daunting. But I think that it's necessary to consider all of these factors when trying to undertake a more mechanistic understanding of the disease. So, what do we know about kind of primary drivers of hydrocephalus? Well, we know that there's a number of genetic causes, and I've contributed to this literature, and then also my mentor, Dr. Hale, where a number of human genetic, unbiased human genetic screens have identified a number of genes and pathways that converge on regulation of the neural stem cell compartment as key pathologic drivers of hydrocephalus. And these studies have relied on kind of aggregating large cohorts of patients and their families and performing either genetic sequencing or genotyping based arrays to identify genes and mutations that are responsible for the disease. And to date, we've identified over 35 genes that have been implicated in primary genetic drivers as primary genetic drivers of hydrocephalus. But what's important is what every single one of these studies has been performed in European populations. And so the extent to which the contribution of genetic forms of hydrocephalus in other parts of the world, like South Africa and Sub-Saharan Africa, are contributing is little known. But I do want to share kind of one recent example of a genetic mutation that causes hydrocephalus that has direct translational impact. And so this was born out of the Nature Neuroscience paper published by Dr. Kale's lab and led by Kyron Dispenza, who's now a neurosurgery resident too. And so what they discovered is that when you took over 2,700 patients and families with hydrocephalus, what they found was the most commonly mutated gene was a gene called P10. And P10 is a tumor suppressor gene that has been very well described in oncology. And it's also been implicated in a number of neurodevelopmental conditions, ranging from focal cortical dysplasias to different tumor syndromes as well. And so what they found was that P10 was the most commonly mutated gene in these genetic forms of hydrocephalus. And what they found, and specifically, was that P10 is expressed very early on in embryology, highest expressed between week 13 to 15 post-affection, and then also enriched in your median ganglionic eminence, which is the kind of stem cell compartment region in the developing brain. And through a series of experiments, what they showed was that these mutations in P10 cause CSF obstruction due to primary overgrowth of the cortex of that gets out either right immediately adjacent to the aqueduct and could cause aqueductal stenosis. But then what's remarkable is that through either genetic suppression of mTOR, downstream mTOR, either through genetic suppression of mTOR or through pharmacological inhibition towards signaling, they could essentially completely rescue the hydrocephalus phenotype in disease. And so that is really the first demonstration that a drug without surgery can completely ameliorate the hydrocephalus phenotype. So at least now this demonstrates some proof of concept that pharmacologic therapy could one day be used to boost the disorder. But it's important to understand that there are many, many, many different mutations in genes that cause hydrocephalus. And here's a schematic from a recent paper we just published on mapping expression of these genes, these hydrocephalus human genetic risk genes would have identified through these screens to the populations in the developing human brain. And what you can see is it's kind of all over the place. You see a lot of genes that are really enriched in the stem cell compartments, but then other genes that are expressed later on in cell types like intermediate progenitor cells, neurons, astrocytes, oligodendrocytes, etc. And so if we start to think about kind of picking off and understanding mechanisms for how each one of these genes contributes to hydrocephalus and all these hydrocephalus, it's become quite a challenging problem. But what we know is that at least for the majority of cases studied, you see that these genes predominantly adhere to affect the neural stem cell compartment, and that will be also kind of a feature that we discuss later with acquired things. So here's these genes that we just mapped to these different single cell populations from the developing human brain. And all of this here is to say that, these genes are expressed in a variety of different cell types. And so by essentially there's a number of different ways that patients can get hydrocephalus as a result of genetic variants on people. But the major kind of concern and the major disease burden that we face in the developing world is due to acquired hydrocephalus. And this is actually the most common cause. Acquired hydrocephalus is the most common cause in the United States as well. So acquired hydrocephalus is not caused by genetic mutations, but rather through downstream perturbation, a signaling cascade triggered by exposure to pathogens or blood. So through detailed in vitro analysis, I will utilize an organoid model called this muscular shortly to kind of understand the pathways that are involved in regulating acquired forms of hydrocephalus with the goal of identifying pathways that may be amenable to pharmacologic changes. And so where does this leave us looking kind of downstream? And do we have kind of a framework to understand, what mechanisms we may look at in many of these models? So we proposed this hallmarks of human hydrocephalus, which is very akin to kind of the hallmarks of cancer, where this serves as kind of a framework or kind of a overarching theme to understand how different either genetic mutations or through brain injuries which may converge on these different features to ultimately cause the disease. So you see here neural stem cell dysfunction, alterations in corticogenesis, brain volume and growth perturbation, defects in synaptogenesis, biomechanical disruption, such things. But what's clear is that much of the work that has gone into kind of define these hallmarks and to try to understand the pathophysiological basis of the disease has been completed in non-human models. So in particular, mice and zebrafish. But we know that understanding human disease necessitates study with human tissue. And this is where neurosurgeons, I think, can have a really unique impact, because our ability to see these patients every day, but also to obtain higher specimens from the population. So to begin to try to kind of tackle and see whether or not all of these hallmarks are recapitulated in human forms of hydrocephalus, we took a simple observation in the OO. So this is a flexible endoscope that has a camera on the end of it and a light source on the end of it. And it can be used to treat hydrocephalus. So you introduce that flexible endoscope into the ventricle, you can look around, you can perform the endoscopic cystostomy, you can even resect, cysts, colored cysts, you do tumor biopsies through the scope. But what's really cool about the scope is that it has a working channel. And so before you even can look around and use the endoscope, you have to take out all the tissue that you pass through as you go from the cortical surface all the way down into the ventricle. So essentially, when you before you can introduce any instruments down the working channel, you get this plug of brain. And so, why not study the primary human brain? And this little plug of tissue is normally just discarded or thrown away. And so initially we put together an NIH grant that was ultimately funded to essentially do functional development funding on tubule. And I'll kind of share a little bit of the data here as we start to think about this problem. So these are our single cell RNA sequencing of human hydrocephalic brain, and we see a really nice representation of many of the major cell types in developing human brains, and astrocytes, neurons, microglia, even very small populations of ocular cells, but also many of the different oligodendrocyte precursor as well as mature oligodendrocytes. And so this is quite a challenging problem actually to start to look at because we're treating children with hydrocephalus, and the pace that at with and the trajectories of the way that all of the different cells in the brain are formed is 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 highly variable. So even if you take a patient that's 4 months old versus 6 month old, those are very, very different in developmental time. And so there's been a number of different atlases that have been published so we can try to understand, here's what we're seeing in our patient with hydrocephalus due to the disease. Is this, just recapitulating development that we would have seen otherwise? Or, essentially, how do we really have a good control? And so what you can see here is that across individual patients with hydrocephalus, you can see a markedly different single cell landscape. And so not only do you have to account for the age of the patient, but also disease etiology and really start to pick this out, one patient at a time. But what came of this initially, which I'll share that we're about ready to getting ready to publish, is we took patients who have acquired hydrocephalus secondary to premature intraventricular hemorrhage. And premature or IVA to prematurity is the most common form of acquired hydrocephalus in developed. And so what we can see here is a number of different things. So in our pre-IVA brains, these children have less astrocytes. They have more microglia, which was expected, just based on an inflammatory cell of the brain. You had decreased total number of neurons, which is not surprising given the other kind of concomitant neurodevelopmental phenotypes you see in these papers. But then what's most striking is that they have this massive accumulation of these oligodendrocytes, all of these different lineages, 1, 2, and 3, with a relatively unchanged pool of the oligodendrocyte precursor cells. But this is a paradoxical observation because we know that the brains of these children are severely undermyelinated. So do we just have a compensatory increase in the number of oligodendrocytes that are around because they can't ultimately move myelin? Are these cells not functional? And we're starting to kind of tackle this question. And so, I'll kind of return to kind of what ultimately prompted the reason I'm talking to you today and missing brain and my work here in South Africa is that we published this initial human genetic study of hydrocephalus, and our top risk gene was actually very phenotypic. It was one of the most human specific genes in the genome. And both the structural the structure of the gene as well as the sequence variations, even between mice and humans, was completely different. And we couldn't use kind of a lower, model organism to understand the impact of our lead candidate genome. And that really necessitated a human system. So, I was starting to think about this and, in speaking with Graham Fegan and what the Neuroscience Institute and at the University of Cape Town was trying to put together. And they had just started this brain organoid program. And so I'm working in the lab of a stem cell biologist named Mubin Ghulam, who trained right down the lab bench from Madeline Lancaster, who was the first person kind of really standardize and describe a lot of the cortical cerebral organoid techniques. And so here you can just see kind of a schematic of how to generate these organoids. And I think what's important to recognize about these is that they're human. So you can take an individual patient and isolate and derive and use pluripotent stem cells, which can then be used to generate different organoid models, ranging from cortical organoids to other cell types in the brain. And using this system, we can introduce either genetic mutation or genetic knockout or genetic variations that we think may play a role in hydrocephalus and study them. And so here's kind of what the organoid looks like in the bin. So these are not, brains in a disc. These are three-dimensional tissues that recapitulate some of the developmental features and transcriptional programs and single cell architecture of the developing brain, but by no means are these brains anymore. But they can recapitulate really nicely some of the layers of the cortex, and they can be a nice experimental tool. And so while understanding the impact of genetic mutation that caused hydrocephalus is important, these are likely to affect the neural stem cell, more, and then would ultimately be best suited by studying these cortical organoid models. But we know that through inflammatory cascades and CSF like secretion, that acquired forms of hydrocephalus that we see in South Africa and continental Africa more broadly are really due to a problem of cerebral fluid inflammation. And so there are techniques to be able to differentiate organoids into ependymal or cord cuts like organoids that can develop these really interesting kind of blood that actually contains CSF like proteins or reminiscent of CSF. And so you can see that compared to a telencephalus control, there are these these colored plexus organoids that develop these kind of CSF like that. And then when you stain so many of the canonical markers of leukoid plexus, you can see them. There's WO1, which is a barrier function protein, as well as PTR and COX-6. And so, like I said, you can isolate this CSF, and it very much resembles um, many of the proteins that we see both in adult, pediatric, and embryonic CSF. So this is a potentially really cool model to be able to understand the impact of brain injuries like infection or intraventricular hemorrhage with precede hydrocephalus. And in addition, they've used this model to understand, permeability of the blood-brain barrier as well. And so, I really think that if we are to understand and try to move hydrocephalus in the direction of a disease which may be amenable to pharmacotherapy, we really need a human-specific model, and we need to be able to look at phenotype that we can assess in a human-specific model like organoids to be able to kind of move that needle. And so here's a nice breakdown kind of the different categories of hydrocephalus ranging from, primary genetic or congenital and acquired causes. And so we're really trying to, link both the kind of the clinical uh, kind of the clinical features of the disease with what we can explain model and get through and understand different molecular and so the entire focus of my time here in South Africa is getting the organoid models here up and running. We're taking iPS cells and deriving iPS cells from a number of different patients of diverse African genetic ancestries here at Red Cross Children's Hospital in Cape Town to be able to try to kind of tackle this problem. But I think more broadly, South Africa is really a country of juxtaposition. And so on the one hand, you see that it's, this beautiful, amazing coastal city with some of the best safari game parks in the world. And, it's frequently named one of the best cities in the world. But then you see this juxtaposition with severe poverty that we see in the town quote. And, highlighted here, and as well as kind of the, recent history of, Nelson Mandela and apartheid, which was only, 30 years ago. But then you also see that South Africa is in many ways is kind of leading the charge with recently they're for the past 2 years, they've been the Rugby World Cup champions, as well as now recently the Cricket World Cup champions. So it's an amazing country with amazing resources, but it's been an absolutely remarkable kind of life-changing experience to be. So kind of, I'm happy to kind of share, what I've been working on, and I'm really grateful to all these funders and different societies that have been critical to enabling this project to go forward. And so I'm happy to take your call. Thank you.
Thank you so much, Andrew. This has been really remarkable. And I am so glad to also point out that we have some of our um, biggest supporters here and the rest of our Mission Brain team. Mr. Uline, thanks for joining us this morning. It's wonderful to see you. It's great to have Dr. Q here as well, who's been commenting on the chat. And we have the rest of our leadership team. JP, Adrian's here, Paula's here, and I think Harsh is here. So if anyone has any questions, please feel free to either put them in the chat. I know some of you are not turning on your cameras because of internet connection. But yeah, again, Andrew, that was wonderful. Thank you for that. Questions?
I just have one comment. Dr. Hale, thank you very much for your presentation. This is amazing. I just wanted to say that I've never seen a basic research project with a global scope, and that is very impressive because um, we have tried to do some things international. Uh, we've done them, but it's very, very hard to communicate to coordinate all these efforts. So I just wanted to thank you and then and congratulate you for your work.
Yeah, absolutely. Thank you. Yeah, I mean, it's probably an entire other talk, and I can just tell you the challenges that I faced over the last 5 months. The U.S. and South Africa essentially have been friends for as long as I can remember, and then the day I got on a plane, Trump got elected, and there's been a lot of you know uh, stress between the two countries. And so that's, severely impacted the way that, UCP has been able to function. For instance, I've been kind of at witnessing kind of the loss of $100 million in NIH sub awards to the University of Cape Town. And so it has completely decimated a lot of the programs here, especially on infectious disease side. And so, I think while I'm in a very privileged position to be able to come do this, and fortunately many of my funding sources were secured before I came over here. I think the real challenge in engaging and doing the type of work moving forward is going to be significant. Hopefully, if the challenges of doing basic science abroad, especially in today's climate, is kind of a whole other topic of, could be a whole other talk.
Yeah, and sharing the samples and all that. Well, thank you and congratulations.
Thank you.
JP, you have a question?
Yeah, and I just want to echo Paula. I mean, what you've done, Dr. Hale, is just amazing what you keep doing. Just have one question. I mean, I've never seen before these organoids for hydrocephalus. I mean, this is super impressive. We do have experience in the lab. Some of our group though organoids with GBM, but I've never seen it for hydrocephalus. And I saw on one of your slides this organoid that with that little bleb that potentially has CSF inside. Are you able to characterize that fluid to confirm it's that like?
Yeah, so you can take a 28 gauge needle and, just under bright field imaging and essentially poke the blood and then, you know, bring it out or draw back the fluid. We can do proteomics and a variety of other things on that tissue. And so that's kind of what we're starting to do now. I mean, obviously it takes a lot of characterization. I kind of glossed over part of it, but really for the acquired form of hydrocephalus, there's a lot of data really to suggest that it's a TLR4 NKCC1, which is a multi-ion channel, and then SPAC kinase, kind of the three player dance where other colleagues' lab has shown that if you can pharmacologically inhibit BAC1 kinase or genetically knock it out, that it completely rescues and ameliorates the CSF hypersecretion and hydrocephalus phenotype. So the eventual goal would be to rely many of the organoid models from IPF cells from many patients of diverse ancestors to be able to understand if an inhibitor for one specific kinase can be effective in all of these different populations. And I didn't have time to really go into it now, but you know, my solo ports paper was entirely in postal series of European ancestry. But when we did the exact same analysis in patients with mixed African ancestry, West African ancestry, mostly that were in the US, we found that many of the genes and the pathways that were associated with hydrocephalus in the mixed African ancestral population was completely different than the ones we saw in the European population. So clearly there is some degree of population specificity, genetic variation background that are contributing to the disease. But I think take take the hydrocephalus stuff completely out of my work. Like ignore that. And if you can just take a single and derive a single IPS line from a patient of African ancestry and put that in global pharma's hands, that would be much more impactful than anything on, because you talk to all these pharma companies, AstraZeneca, all these major companies, and they want, they want to test drugs on non-European ancestry patients. And so, that's kind of something we're talking about now, is figuring out how to embed our lines that we're generating here from at Cape Town into kind of pharma and be able to make them more broad.
That's really impressive. Thanks so much.
Dr. Q, you have your hand up.
Thank you, April. First of all, thank you guys. This is an amazing talk. I came in sort of halfway, and I apologize, Andy. I'm running around the hospital. We had an emergency this morning, but I was very impressed with the data, and I was impressed with your answers. And I thank you immensely for inspiring us to do better. Just, I do agree with Paolo's comments about, doing international work is not easy, and doing it at this caliber with the data that you have presented, it's inspiring, to be honest with you. And I guess my question is to follow up also with JP's comments and question. You're doing amazing work already with the organoids. You're characterizing them. Your answers illustrate that you're thinking further. From our perspective, given the work that we do in Mission Brain that it's got global implications, what can we do to help you continue to further this work and take it to the next level?
Well, thank you so much. I appreciate your kind words and for starting up. I'm very grateful for your support and for Mission Brain support. I mean, I think the frank truth is that this stuff costs a lot of money, and I think that's something that is really hard to kind of grapple with when you're trying to do things at a high level, but in a more resource constrained environment. You often have to do things that are creative to kind of get solutions to be able to move forward. I mean, I think that using kind of the Mission Brain platform to advocate for science funding and diseases that are relevant to global neurosurgery would be incredible. I mean, I think mainly the challenges here are not willingness of people to do the work, but I think it's finding enough funding, honestly, to sustain the programs.
Thank you, Andy. We'll continue to work. I mean, as you know, we are supportive we have extraordinary people like so Richard right here that are not only inspired, that continue to help us, and will continue to help. And thank you, April, also for your leadership. We continue to do what you're doing as well to bring in these speakers to this series. I'm very, very impressed. So thank you. I'm going to sign off. I'm going to go back to the hospital, and thank you for your time and everybody. And I see no other questions. I'll stay for this question and then I'm going to sign off.
All right. Thanks, Dr. Q. Adam, you have a question?
Yes, I do. Can you guys hear me?
Yes, loud and clear. And tell us where you're coming from.
Awesome. Yes. So my name's Adam. I'm a MD PhD student over at the Icahn School of Medicine at Mount Sinai. And I have my essentially preliminary research interests are looking at cerebral small vessel disease, and the etiology of stroke and neurodegeneration. And given your experience, as a neurosurgeon working in the global neurosurgery field, could you comment a little bit on sort of what the field of stroke research looks like on the global scale?
Yeah, I mean, this is certainly not my, my specific area of expertise, but I can kind of share some anecdotes. So, there's an amazing endovascular program here at the University of Cape Town. But I think that when you start to think about, stroke care, it's really more than a network problem. It's a systems problem of being able to identify patients with stroke early, get them to their right, into the right centers, into providers that can ultimately perform, life-saving measures like some. So I think in the global context, there's a lot more challenges in terms of, putting systems in place, who's going to drive the ambulance, who's going to, if there's a scan that shows a likely kind of large vessel occlusion in a hospital that's an hour and a half away from, the mothership, so to speak, at University of Cape Town, how is all of that going to work? And so, I think that for a problem like stroke, I think it's mainly a kind of systems.
Okay, thank you.
Great question, Adam. Any other questions from anyone in the audience? If you're feeling shy or if you can't say it out loud, you can just type it on the chat. But I think, Andrew, we've had some really interesting questions and comments, and it's been really a wonderful lecture and very eye-opening for a lot of us. This lecture has also been recorded, and we hope to be able to share it. Really, I think this is only the first lecture series we're having this year, and it's been really remarkable. Any other questions so far from anyone else? Comments? All right. Well, we'll have 3 more minutes left, and Andrew, I'd like to give the floor to you if you have any comments or suggestions. You know, we've heard you talk about how important some of the support that we provided, and also some of the things. It's great to know also how your experience in South Africa has changed your perspective, if it has, in terms of how you practice moving forward. So I'd take it away.
Yeah. I mean, I think, I think there's a few things. I think, on a personal level, moving to a new place with a new culture, with new setting, new people, always presents some challenges, but also many opportunities. And so, I came in with, honestly pretty burned out from junior residency, and, trying to get things moving and kind of seeing how things are going. And, when you start doing new things, with new people, you also have to kind of take the time to be able to be patient. And I think that, learning that patience in this setting has been really good for me, because I think that, in the U.S., especially U.S. neurosurgeons, there's, a tendency to go, and, everything is tied to your productivity. And if you lose an hour, you lost so much time, and, all these things. And then you realize that all of the things that, would get under your skin in terms of, delays, thinking about, your case gets delayed, or, you're up late taking call, like all those sorts of problems become and really minimize the few other people across the world do things, and that everyone always has their own unique problems. But I think that um, that it's it's a really kind of amazing opportunity to be able to see how other people do things. And so, yeah, if anyone is interested in doing any sort of kind of basic science or basic research abroad, I'd be happy to talk with them. I think there's a number of challenges, but I think that they're not that they can be overcome.
Wonderful. Thank you so much. I'd like to thank our team who's been organizing this event. Brooke, thank you so much for doing all the work behind the scenes. And I see Adrian and Harsh as well, and JP. So in behalf of Mission Brain, thank you very much again, Dr. Hale. Thank you all for coming today. And hopefully we will see you again in the next lecture series, and we'll send out the schedule for that. Uh, Mr. Uline, do you have any last words to say, or you? Wonderful. We'll see you. We hope to see you in the next one as well. Take care, everyone. Enjoy the rest of your weekend. Bye bye.
Thank you.